Those are the letters of genetic code, the 25,000 genes you've got written in your DNA. This is what they typically talk about, the genetic code. This is what they're talking about. But I want to talk about a different aspect of DNA science, and that is the physical nature of DNA. And it's these two strands that run in opposite directions for reasons I can't go into right now, but they physically run in opposite directions, which creates a number of complications for your living cells, as you're about to see, most particularly when DNA is being copied. And so what I'm about to show you is an accurate representation of the actual DNA replication machine that's occurring right now inside your body, at least 2002 biology. So DNA is entering the production line from the left-hand side, and it hits this collection, this miniature biochemical machines that are pulling apart the DNA strand and making an exact copy. So DNA comes in, it hits this blue donut-shaped structure, and it's ripped apart into its two strands. One strand can be copied directly, and it can be seen spooling off down to the bottom there. But things aren't so simple for the other strand because it must be copied backwards. So it's thrown out repeatedly in these loops and copied one section at a time, creating two new DNA molecules. Now, you have billions of this machine right now whirring away inside you, copying your DNA with exquisite fidelity. It's an accurate representation, and it's pretty much at the correct speed for what it's occurring inside you. But I've left out error correction and a bunch of other things. This was work from a number of years ago. Thank you. This is work from a number of years ago, but what I want to show you next is updated science. It's updated technology. So again, we begin with DNA, and it's jiggling and wiggling there because of the surrounding soup of molecules, which I've stripped away so you can see something. DNA is about 2 nanometers across, which is really quite tiny. But in each one of your cells, each strand of DNA is about 30 to 40 million nanometers long. So to keep the DNA organized, to regulate access to the genetic code, it's wrapped around these purple proteins. I've labeled them purple here. It's packaged up and bundled up. All of this field of view is a single strand of DNA. This huge package of DNA is called a chromosome. And we'll come back to chromosomes in a minute. We're pulling out, we're zooming out through a nuclear pore, which is sort of the gateway to this compartment that holds all the DNA called the nucleus. All of this field of view is about a semester's worth of biology, and I've got 7 minutes, so we're not going to be able to do that today. No, I'm being told no. This is the way a living cell looks down a light microscope, and it's been filmed under time-lapse, which is why you can see it moving. The nuclear envelope breaks down. These sausage-shaped things are the chromosomes, and we'll focus on them. They go through this very striking motion that is focused on these little red spots. When the cell feels it's ready to go, it rips apart the chromosome. One set of DNA goes to one side, the other side gets the other set of DNA, identical copies of DNA, and then the cell splits down the middle. And again, you have billions of cells undergoing this process right now inside of you. Now we're going to rewind and just focus on the chromosomes and look at its structure and describe it. So again, here we are at that equator moment. The chromosomes line up, and if we isolate just one chromosome, we're going to pull it out and have a look at its structure. So this is one of the biggest molecular structures that you have, at least as far as we've discovered so far, inside of us. So this is a single chromosome, and you have 2 strands of DNA in each chromosome. One is bundled up into one sausage. The other strand is bundled up into the other sausage. These things that look like whiskers that are sticking out from either side are the dynamic scaffolding of the cell. They're called microtubules, but the name's not so important. But what we're going to focus on is this red region. I've labeled it red here. And it's the interface between the dynamic scaffolding and the chromosomes. It is obviously central to the movement of the chromosomes, but we have no idea really as to how it's achieving that movement. We've been studying this thing called the kinetochore for over 100 years with intense study, and we're still just beginning to discover what it's all about. It is made up of about 200 different types of proteins, thousands of proteins in total. It is a signal broadcasting system. It broadcasts through chemical signals, telling the rest of the cell when it's ready, when it feels that everything is aligned and ready to go for the separation of the chromosomes. It is able to couple onto the growing and shrinking microtubules. It's transiently, it's involved with the growing of the microtubules, and it's able to transiently couple onto them. It's also a tension sensing system. It's able to feel when the cell is ready, when the chromosome is correctly positioned. It's turning green here because it feels that everything is just right. And you'll see that there's one little last bit that's still remaining red. And it's walked away down the microtubules. That is the signal broadcasting system sending out the stop signal, and it's walked away. I mean, it's that mechanical. It's molecular clockwork. This is how you work at the molecular scale. So with a little bit of molecular eye candy, we've got kinesins, which are the orange ones. They're little molecular courier molecules walking one way. And here are the dynein. They're carrying that broadcasting system, and they've got their long legs so they can step around obstacles and so on. So again, this is all derived accurately from the science. The problem is we can't show it to you any other way. Exploring at the frontier of science, at the frontier of human understanding, is mind-blowing. Discovering this stuff is certainly a pleasurable incentive to work in science. But most medical researchers, this is just discovering this stuff is simply steps along the path to the big goals, which are to eradicate disease, to eliminate the suffering and the misery that disease causes, and to lift people out of poverty. Thank you.
So, like, that's just so ridiculously mind-blowing that it's almost unbearable. I mean, to think about that as clockwork even is a pretty strange idea, because, well, those little things walk over obstacles. It's like, how the hell does that happen? They're just molecules. So it's so cool, because when you go down, you'd think simple, but, you know. And, you know, he said at the beginning when they were taking that, when the little machines were taking that DNA apart, that he didn't show the error correcting. You know, there's other little machines that go along and see if everything's okay, and if it isn't, they cut it out and put a right piece in. It's like, yeah, things we don't understand. There's no shortage of them, that's for sure. Okay, so what I'm doing in some sense is walking you through a psychophysiological or representation of Piaget's developmental process, I would say. So I wanted to zero in on the hypothalamus because it seems to me the thing that sets the most basic frames. And so we'll go ahead with that. So you see that it's made up of all these little parts. And so it's called the hypothalamus more for convenience than because it's a homogeneous set of structures, because it's not a homogeneous set of structures. And this is something to consider very carefully when you're thinking about the terminology that psychologists use or that you might use to describe your own behavior. Because, you know, you can roughly, there is a psychology of motivation and there's a psychology of emotion. And you might think, well, emotion and motivation are categorically different entities. But they're not. In fact, there's no such thing as a uniform set of motivations, and there's no such thing as a uniform set of emotions, and the distinction between a motivation and an emotion is unclear, to say the least. And that's partly because the physiological substructures that subsume what we call motivations and what we call emotions, and it's not like there's a motivation center that's homogeneous. The closest is the hypothalamus, but it's made of structures that are qualitatively different. And then the emotions, because I have to use that descriptive terminology because we have to communicate about it somehow. There's all sorts of different structures in the brain that contribute to emotional expression, and they're not even in the same place, much less composed of identical structure or function. So, you know, we have these shorthands that we use to divide up the world, but they're awkward and untenable as the level of resolution increases. But anyways, I'm still going to go with motivation and emotion because it's a useful simplification, but you can see with the hypothalamus that there's all these, you know, complicated little subsystems in there. And then I showed you that video to show you just how complicated the subsystems are, all the way down to the, really to the molecular level. How those little machines manage what they do is completely beyond me. You know, to call it clockwork when those little things that walk can walk over obstacles. It's like clockwork does one thing, you know, only. Click, 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 click. That's all it does. No exceptions. This thing walks over obstacles to get where it's going. It's like, who knows what's going on down there. But it works well enough, so here we are, weirdly enough. So, motivation seems to me to be the initial framing process. And you come into the world with the motivational systems roughly ready to go. You know, babies are hungry, babies get cold, babies want something to drink. You know, so the world already comes, in some sense, they come into the world with pre-packaged categories for existence, and those are the categories that are going to aid their survival. And, you know, they're not simple either. It's not so simple. It's, well, hunger, thirst, pleasure, pain, anxiety, you know, the classic sort of, and the classic emotions, sadness, joy, and so forth. Those systems are already there, but babies have more complicated systems too, like the system for exploration is already in place, and the system for play, which is really complicated, it's already in place. And so, you know, you come into the world with a human nature, and the nature seems to be distributed across these subsystems. That's one way of thinking about it. And it's also useful to think about the operation of those subsystems as something like, you could think about them as games with an aim. You could think about them as stories. You could think about them as frames of reference. You can think about them as action patterns. All of those, and you can think about them as sub-personalities, which I actually think is maybe the best way to think about them. Because if you're hungry, it's not a deterministic drive. It's a sub-personality that has a goal, and then it has a bunch of action patterns that are going to work in reference to that goal. It has a bunch of perceptions that suit that goal, and it organizes your emotional responses around that goal. And so to think about it as a personality is it's a much more intelligent way to look at it. One of the things about Skinner's rats, you know, Skinner could get rats to do almost everything, and he would reward them with food. And so he had a simple rat model, but his rats were starved down to 75% of their normal body weight. So not only were they not social, gregarious rats like rats are, because they were isolated, they were genetically altered from wild rats, but they also weren't as complex as a real rat because they were starving. And so, but you know, a starving rat is a pretty good model of a rat, and a rat is a pretty good model of a person. But a lot of our models of simple behavioral learning were based on starving, isolated rats. So anyways, how to think about motivation? We'll think about it from the hypothalamic perspective. So we could say one thing that motivation does is set goals. We could say that emotions track progress towards goals. And I'm going to use that schema even though it's not exactly right. So you say, well, motivation determines where you're going to aim. So if you're hungry, you're going to aim at something to eat. And then that will organize your perceptions so that you zero out everything that isn't relevant to that task, which is almost everything. You concentrate on those few things that are going to facilitate your movement forward. When you encounter those things, that produces positive emotion as you move through the world towards your goal, and you see that things are laying themselves out that facilitate your movement forward. Those things cause positive emotion. And if you encounter anything that gets in the way, then that produces negative emotion, and it can be like threat, because you're not supposed to encounter something that gets in the way. It can be anger, so that you move it away. It can be frustration, disappointment, grief. Those would, if you had a response that serious to an obstacle, it would probably punish the little motivated frame right out of existence. You know, so you walk downstairs and, I don't know, the contracting company has set a wrecking ball through your kitchen. It's like, that's going to be disappointing. You're not going to keep eating the peanut butter sandwich in the rubble. That little frame is going to get punished out of existence, and some new goal is going to pop up in its stead. And you know, one of the things we're going to try to sort out is how do you decide when you've encountered an obstacle that's so big that you should just quit and go do something else, because that's not obvious. You know, and you can get into counterproductive persistence pretty easily. So we don't know how people solve that problem. It's a really complicated one. So anyways, we're going to work on that scenario. Your hypothalamus pops up micro goals that are directly relevant to biological survival. That produces a frame of reference. So it's not a goal, it's not a drive, and it's not a collection of behaviors. It's a little personality. And the personality has a viewpoint, it has thoughts that go along with it. It has perceptions, it has action tendencies, all of that. You can see this in addiction, most particularly. So one of the things that you find often with people who are alcoholic is they lie all the time. And that's because when they've built a little alcohol-dependent personality inside of themselves, or a big one, maybe it's 90% of their personality. And one of the things that consists of is all the rationalizations that they've used over the years to justify their addiction to themselves and to other people. And so the addiction has a personality. You know, and so when the person is off, or maybe they're addicted to meth or something like that, where, you know, the addiction is more, it's more short-term powerful, than I would say, than an alcohol addiction. They'll say anything. And the words are just tools used to get towards the goal. And if they happen to be deceptive, whatever, it doesn't matter. They're just practical tools to get towards the goal. And then when you get towards the goal and you take a nice shot of meth or something like that, you reinforce all those rationales that you use to get the drug, and then the next time you're even a better deceiver and liar. So, okay, so we're going to say motivations One way of thinking about it is they set goals, but it's not the right way of thinking about it. They produce a whole framework of interpretation. And so we're going to think about that framework of interpretation. And then emotions emerge inside of that.